Gives me great pleasure to introduce Sarah Johnson. Sarah did her undergraduate work at Washington Uni and got a degree in maths. Then, for a change, went to the UK and got a second BA in philosophy, politics and economics. Then a master's in biology at Oxford, followed by a PhD with Maria Zubrin at MIT in planetary science. The title of her thesis was Mars in the Late Noachian Evolution of a Habitable Environment. Sarah's been in Georgetown now for 18 months and a member of the Sample Analysis at Mars team, the SAM team, on Curiosity since last summer. The title of her talk today is The Search for Life on Mars and Icy Worlds, Insights from Molecular Biology.
Thank you. Can everybody hear me?
Yes.
Okay, great. Well, thanks so much, Julie. This is such a pleasure to be here. I'm a new assistant professor down at Georgetown, and I'm a planetary scientist and an astrobiologist, but we don't have a department of planetary science at Georgetown, and so I've been spending a lot of time with the Planetary Environments Lab out at Goddard, and I just love the work that Carnegie is doing in planetary science as well. I adore my new job, but one thing I do miss is having planetary science colleagues to talk about all of these fun ideas with. So anyway, it's just such a pleasure to be here and to meet so many of you today and hopefully talk to some other folks over lunch as well. But I thought I would just give you a quick talk about some of the stuff that's kind of been going on in my head and some of the things I'm trying to get started with my fledgling new lab down at Georgetown. And I thought I'd really try to just focus in on three things. I'm hoping, my vision, is that one day I'll have a molecular biology wing and an organic geochemistry wing, and we'll all be thinking about these ideas with biosignature detection in planetary environments. The molecular biology stuff is further along, so I thought I would talk about that today and focus on, just give you an example of a couple of the projects that I think that we can use the tools from molecular biology to help us better understand what's going on and how biology is affecting patterns of mineralization, particularly in Mars analog environments, and some of the things we might be able to use by applying metagenomics and transcriptomics to these environments on Earth that may help us better interpret the data that we're getting back from other planetary places. And then I love these third-generation sequencing approaches. So the world of genomics has just been exploding over the last 5 years, and we're now moving from next-generation sequencing into third-generation sequencing. So I thought I would tell you a little bit about that in the context of a recent field season that I had in Antarctica, and then finally end on possibilities for life detection that sequencing might offer, including a possibility to detect life that's not actually DNA or RNA based, so still harnessing the power of sequencing. So we'll get to that mystery here in just a minute. But this is just sort of a slide showing all of the advances that we've had in the world of biology. I'm sure everyone has observed this incredible thing with the Human Genome Project, and then moving from the Sanger sequencing into these next generation sequencing platforms, which is really the mainstay of all the sequencing cores that are at different universities and different labs around the country now. And then slowly we're also now entering this third generation sequencing. And I'll get more to this in a minute, but this is sort of where you're looking at single molecules of DNA, sequencing native DNA as opposed to PCR amplicons. But using kind of these next generation sequencing approaches, I'll talk about that first. So there are these questions that you can ask. You can start off with a microbial community, and you can ask the sort of very simple question, what microbes are there? And this is looking at 16S or 18S sequencing, and this is the sort of most simplistic thing. This is what a lot of studies were based on just 5 years ago. And this is looking at one very highly conserved gene within a microbe, and just by sequencing this very small gene, you can figure out very well, because it's so highly conserved, where this sort of falls on a phylogenetic tree. And you can do an identification. You can say, okay, well, this is E. coli, or this is Micrococcus, or this is these types of organisms, and you can do some nice diversity plots and see kind of who's there in your community to start with. But you can also move to the next step, where you're asking, what's the genetic potential? And that's using metagenomics, where you're looking at the entire gene, the whole genome, not just that one very highly conserved gene. And so this will start to tell you some things about what's the metabolic potential. What sort of machinery is there in that particular snapshot of your environment, this microbial community, what are they capable of? Are they doing sulfur oxidation? Are they doing iron reduction? You know, what kinds of things do they have the molecular machinery for? But of course, just because you have a gene, you may not be using it. And so you can move on to this final question of kind of what are these microbes actually doing? And so metatranscriptomics is now looking at the gene expression. So looking at the RNA and the gene expression that's actually happening. So that's looking at RNA. And then metaproteomics is when you start looking at peptides. So now you've got the RNA there, but what is that actually encoding for? And are those peptides actually being made? And so those are the next two steps that you can go down this process. And so using these sorts of approaches, I've joined this research collaboration in the last year, which has been super fun. And it's applying these kinds of techniques to the extreme microbiome, to extremophiles that are here on our planet. And this is a great collaboration of a bunch of folks, some in astrobiology, some more kind of just on the extremophiles side of the house, interested in asking, you know, what are going on in these communities. And this is some sample. You can see these gypsum crystals. This is a place in Australia, which I'll talk about momentarily. But it's great. I mean, these people that work on this is this doorway to hell. I don't know if anybody's heard of this. Off in Kazakhstan, this thing's been on fire for 30 years, and folks are rappelling in and grabbing soils and trying to find. So it's like very extreme, right? But lots of different environments around the world. So we have these conference calls, and one of the things that we've been really working on is it's tough to study extremophiles. So we have these more traditional methods that we have where we look, you know, microscopy, and we look under a microscope and we see, oh, okay, that looks like this type of organism. And then we also have these culturing methods where you put something you collect from an environment into media and you grow it up. But studies suggest that, you know, up to 99% of what's actually out there won't grow once you pull that back into the laboratory setting, or won't grow in pure culture at least. But then when you have these metagenomic analyses, you get a bigger swath of the puzzle. But one of the things that's tricky about organisms, especially organisms that are adapted to live in tough conditions, are that just because you get your DNA out of your sample, it doesn't mean you're actually seeing everything that's there. Because you can get this bias, because the easiest to lyse cells are going to be the ones that you're going to get the DNA from. And if you've got really difficult recalcitrant spores, you might not be breaking them open. Yeah.
What does the word lyse mean?
Lyse is to break open a cell. So yeah, no problem, sorry. If you have any questions, please feel free to interrupt at any time. So one thing that I've been using lately, which I really love, is this microbial standard. So these are known cell counts of these particular 10 things that vary widely in gram stain and GC content. And you know, you've got actia, you've got bacteria, and we are spiking our extractions now with this mix. And if we don't get all of these things back using the extraction method that we're using, then we know that we've got bias in our sample. And so that's something that really hasn't been done widely. And I think it's super important just to make sure that we're actually seeing everything that we've got. And then we've also been developing these new methods that you are, instead of using, say, one enzyme or just one physical method to try to lyse your cells, this is a polyenzyme cocktail where you've got 6 different things and you attack the cell with all 6 of them all at once. And you can sort of see, even just looking at this, so you can see like these cells, there are major changes after you sort of put the polyenzyme in. And so these are micrococcus cells, and you can see these cell walls in these cells. And if you slowly watch as the digestion proceeds, you can see these cell walls break down, and then these spiroplasts form. So this is like the cell membrane now breaking out of this cell wall, becoming in these much more spherical shapes. And then you hit them with a little bit of detergent and pop. You've got this stuff now. All of these cell walls are broken, and you're really getting in to see a lot of what's going on. And this is just, I had my postdoc put this together from a recent Antarctic sample that we were working at. So these are the standard mobile soil kits that you can buy off the shelf, and you can do a quick soil lysis. But you can see that once you start adding this polyzyme, your yields go up dramatically in terms of how much DNA you can recover from your sample. So I've been trying to apply some of these new methods as well as this metagenomic, metatranscriptomic approach to studying some of these Mars analog environments. And I thought I would dive a little deeper into this one particular one, these acid salt lakes that are out in Western Australia. So if you fly over to Perth and you rent a truck and you drive like 14 hours back into the desert, you can get off into this beautiful area, which is right at the edge of the Nullarbor Plain. And it's in some of the oldest rocks we've got here on Earth. You know, here these are Archean rocks, and the Yilgarn Craton, so fly in there and go like there. And just to give you a sense of the geologic setting, so these are ephemeral playa lakes that are incredibly salty and in some cases very, very acidic. And they are very small, some of them just meters in scale, some of them are larger, kilometers in scale. And you've got this really interesting mix of minerals. So you've got gypsum and halite and alunite, but you've got the iron oxides, jarosite, which those of you that study Mars know has been a sort of a traditional indicator of these incredibly acidic conditions on Mars detected in places like Meridiani Planum, kaolinite. But also, so you start getting these clays, you see kaolinite and smectite, and just, I'll get to that in a second. But you can see these things are kind of wild looking and really, really salty, and then hosted by these red beds. You've got some felsic rocks there, but you've also got mafic and ultramafic rocks cutting through as greenstone belts. And the sense of the mineralogy that you see there is in many ways similar to the sorts of detections that we are finding on Mars, in places like this. And so one of the reasons that we decided to go to Gale Crater was because there was an interesting mix of sulfates and phyllosilicates. So phyllosilicates or clays are typically thought to form in kind of more neutral pH conditions. And sulfates, like especially these acid sulfates, like the jarosite, are thought to form in much more acidic conditions, you know, say pH 2 or less. And we have this whole paleoclimate theory for Mars, the beef ring theory that, you know, says first billion years, warm, wet, nice clays. And then next billion years, acid, arid, acidic, you get these acid sulfates. And basically you see the clays on lower terrains, you see the acid sulfates on higher terrains, we have our stratigraphic superposition, that's how we get to this theory. And I think that, you know, largely that makes sense, but we've got places like Gale Crater, where we see these interbedded sulfates and phyllosilicates, and in some cases mixed. And it's interesting because, you know, how did these minerals form? And are these different climatic episodes, or is there a possibility that maybe some local geochemical changes could lead to these things forming together? And, you know, we've just started our ascent of Mount Sharp, and so we will be looking, you know, over the next couple of years in much more detail into some of these different units. And we've, of course, been down in the base of Gale Crater exploring lots of clays and mudstones. So I think it will be very, very interesting. But one thing that we can do, so here's what I was saying. So this is just one of Ralph Milliken's kind of first images that really convinced folks to go to Gale Crater. You know, you see this really nice layering in the lower base of the mountain. And this is an image from a colleague, Alice Baldridge, where you see it's a different color scheme here. We've got cyan and we've got yellow, but showing these mixed clays and phyllosilicates. And this is from Australia, from hyperspectral airborne imagery. And these are inner dune deposits between these playa lakes, where you see this very interesting mix where you're getting phyllosilicates and sulfates very closely together in terms of lateral extent.
Sorry, excuse me. What do the colors represent again? Is it compositional or?
Yeah, so it's the sulfates and the phyllosilicate units, and then there's also I think there's also iron oxides in here, which are the greens, but the sort of cyan and the yellow. So you can see that we've got again these two large, you know, classes of minerals, the sulfates and phyllosilicates, sort of again kind of interbedded, showing up in these kind of layers in these inner dune deposits. here in Australia. So this is a smaller scale too also. This is a 1/4 kilometer. And so there was a study by Stacy Story that was published in JGR where she looked at cores going back in time and did see again these same sort of minerals that I mentioned before, the gypsums, the jarosites, the alunites, and then these smectites as well, which is again very interesting. You know, one of the things that I think has been really neat too is Doug Ming down at Johnson Space Center has been doing experiments on acid smectites and actually been able to form some of these smectite materials, which again we often think of as neutral pH materials, if not slightly elevated, under more acidic conditions. Another great thing about this site is you see these sedimentary features that in many ways are similar to Mars. So in these on the left are all images from Mars, from Meridiani Planum, and so you get these large crossbeds, you get mud cracks, you get these crystalline vug structures where you can get a crystal forming and then disappearing as we see here in these vugs, and you see them in situ in these Australian examples. So all in all, it's a nice analog to work with. It's not perfect, but it's a good place to sort of start testing these theories of, well, there is biology in this system, and is it having an impact on sort of how the mineralization is occurring? So this is another example of just kind of how quickly this field has progressed. So there was a great study done by Melanie Moore-Mile, the first study of the kind of astrobiology of these lakes. And so this was just a couple of years ago. But you get, you know, your 24 sequences. This was a clone library that you put together, and you know, you find out, okay, well here's a snapshot of some of the different organisms that are in this environment. And then just to give you a sense of how quickly the field has changed, so just in the last, you know, 3 or 4 or 5 years, we've really been trying to embrace more of these high throughput methods. And so a study of the metagenomics of this system, now you can look at, say, 2 million sequences or 4 million sequences, and you can get a lot more information. And again, instead of just knowing from the 16S gene, you know, who's there, we can now start asking questions about what's the metabolic potential of this environment. And so if you look on that entire, all of those, across all of the DNA, across the entire genome of this environment, you get a lot of genes that are associated with sulfur metabolism, including a ton that are associated with this particular pathway called the SOX pathway, or the sulfur oxidation pathway. And you get this in photosynthetic, but as well as non-photosynthetic sulfur oxidizing bacteria. And you can get thiosulfate oxidase to sulfate. You get these series of SOX genes, and we see these genes in high abundances, you know, sort of in this environment. And one of the things it tells us is that this microbial activity through these gene pathways may be generating a considerable amount of acidity right there, and it could be altering local geochemical conditions just because of the biological activity happening. And this raises a lot of really interesting questions, and this is a piece of ongoing work, partly motivated by this neat study that Kelsey Norland also did a few years ago. So one of the big players when we look into this community is a bug called Acidify Bacillus. And so this is doing sulfur oxidation, you know, it kind of in that lake sediment. And so we cross from the aerobic to the anaerobic boundary right there in those lake sediments. And one of the things that's interesting is, so we think of jarosite formation. If it's a completely abiotic process, again, it has these very stringent limitations for what the pH is. It only happens under very, very acidic conditions. And also, when you think about water limiting positions. So we can get abiotic microbial jarosite sort of happening here up in this one little quadrant of the triangle here. But once you take biology and involve it, so these were laboratory-based studies using an Acidify Bacillus and showing that you can actually precipitate that jarosite as a product of the metabolic activity of this organism under a much wider range of conditions in terms of less oxidizing, less water limiting, and higher pH. And so one of the things that's interesting about this is when we often look through this lens of, sort of an abiotic world, if we're looking up at Mars, we may say, well, Gerasite, this must be super acidic conditions. But what may be also happening is that if microbial biology is involved, you can have much wider, sort of a bigger lens, a wider horizon. And so we're doing some ongoing work here, starting to bring in some sulfur isotopes as well. But I just wanted to tell you about that study and then two others that are quickly going on, just thinking, again, along these lines of how biology may be affecting patterns of mineralization. So a similar sort of thing here where you get a lot of sulfate deposits in these lava tubes. And so this is a new project for the lab working in Hawaii as well as in Idaho at Craters of the Moon, where you get very high iron content basalt, another nice analog for some of the basaltic lava flows on Mars. Lava tubes are great because they're near surface, but they're thick enough that you could have some significant radiation protection. And you might even be able to kind of climb down with a cliff thought one day and explore some of these near surface lava tubes. And there are a lot more of them on Mars than there are on Earth. And they're bigger because there is less gravity, but they're associated with these kind of shield volcanoes. So you get these beautiful images. This is a LiDAR image sort of showing there are little people on the surface, and here's the lava tube underneath, and they sort of collapse down. But there's a theory that was put forth by a guy named Doc Richardson a couple of years back that these sulfates are microbially mediated, and that you get these particular organisms as a byproduct of, again, sulfur oxidation in the roof of this lava tube. And the way this theory was formed was by using spectroscopy and looking and finding that there were these mass defects within the structure that looked like they would potentially hold organic residues. But this is also kind of one of these questions. This reminds me of some of the arguments that I have with my father when we say, who was president in 1928? And you could argue, or you could just pull out your iPhone and look. And we're sort of at this point with these metagenomic and transcriptomic studies. You can just kind of look, and you can see it's really quite easy to just go in and say, well, are these actually microbially mediated? Like, are we seeing organisms there that have this capability, and are we seeing the gene expression that would be associated with actually precipitating these minerals? And so this is an ongoing study that we've got going on. Actually collected samples of these, threw them into a liquid nitrogen prime cryoshipper at minus 190 degrees Celsius, which completely freezes time. And then we can go back and we can look at these transcripts and we can see, do we see gene expression associated with this? And my postdoc is working on this right now, but we've got some cool initial results there. And then one last one I wanted to tell you about is an upcoming trip that we're going to do in June. One of the most intriguing things that came out of the Spirit Rover investigation at Gusev Crater were these little nodular silica center features that were discovered in 2008. And then shortly thereafter, in 2009 in April, Spirit had moved on and then got stuck in some sand and was pointed the wrong direction. And the poor Spirit Rover lost energy because the solar panels were pointed in the wrong way and couldn't power up and couldn't heat up and died. So we actually had the wrong theory for how these formed. We thought there was kind of this acidic fumarole sort of thing going on. But on further study, especially looking at the spectra coming off of these features, it seems really clear that this is our first in situ example of a hydrothermal site on another planet. Here on Mars. We've got this evidence that these were formed. There was this silica that was discovered, soils over 90% of weight percent silica, and the phase was opal A, and there was a thin little halite crust on top of these. And so these pictures are from Mars. And one of the really neat things about it was there were these weird kind of microdigitate structures here. And so this was a big mystery for a while. And then Steve Ruff and Jack Farmer, who are folks out at Arizona State, they went and they found this analog environment. So these are the pictures over here. And this is the El Tatio geyser field, which is in Patagonia. And it's really high up. It's in the Atacama Desert. And it's spectrally a near perfect match where you get this opaline silica, very, very thin halite coating, and these really weird nodular features, but also some of them have these microdigitate, kind of these little fingers kind of pointing out. And so they published a recent Nature Communications paper about this, but using microscopy suggested that the ones that are more nodular are not the same kind of, if you cut these open and you use SEM, you can see these calithrix, these cells that look like they're lining up, making microstromatolites, and that these would be microbially mediated. Potentially, this particular morphology could be specifically microbially mediated, whereas these ones are less so, and these ones have a clear kind of microbial mediation component. And this has actually become something rather important because we're in the landing site down select process for the new Mars rover, the Mars 2020 rover, which is really, I think, going to determine more than even the next decade of Mars exploration. This is our last sort of big flagship that we've got on the books, and the idea is to cache samples at this place and then bring them back to Earth to look for signs of biosignatures. So we're finally moving from searching for water, searching for habitability, into actually searching for signs of life, with the hope of bringing these samples back to look at in Earth laboratories. Do we have the mission yet to do that? No. But that mission has to go back to the same place to pick up those samples and bring them back. So we're looking at putting a lot of eggs in the same basket for this next Mars mission that we choose. And we're down to 3 different landing sites. One is Jezero Crater, a delta. One is the Columbia Hills, going back to where the Spirit rover found these precise deposits, and the other is northeast Syrtis. So we're going to pick one of these landing sites, and this is on the table, the idea of going back to Gusev Crater to look at this, and primarily on the strength of this idea that this is our only hydrothermal site that we know of for sure in situ that we've seen before, and we could potentially bring back this exact stuff, and it could have biosignatures in it. So I'm very excited. We're going to go down with them in June, and we're going to do the same sort of thing. We're going to, instead of just taking these microscopy approaches, expanding that and looking at, what actually are these microbes and what's the gene expression associated with them. Okay, so then this was also super fun. This was an Antarctic field season that we just got back from about 6 weeks ago, something like that. And so this is cool. These are these perch deltas that are in the dry valleys, and we were being super clean, and we were trying to take these samples, and we were actually, the thing that we were doing was a little different. So I'm not going to focus too much on, there's another aspect of this study, which I think you guys will find more interesting. But one of the things that's tricky about doing genomics in a really cold, arid environment is that you have this incredible preservation potential. So if you're going to go looking for life, and if you're going to be looking for ancient life, you're going to look for lipids. We have lipid biomarkers that are billions of years old that we found here on the Earth. Nucleic acids do not stick around very long. If you're out in the tropical rainforest, your cell dies, it is gone. Your nucleic acids are not going to be there very soon after. But if you go to a place that's incredibly cold and incredibly dry and salty, you can get this really high preservation potential. That's really interesting. So this is where all of these studies beforehand are focusing again on these next generation, the mainstay sequences. We use sequencing technique. We use an Illumina platform. And the way the Illumina platforms work are you get these very short reads, but they're very high accuracy, but they're 150 base pairs to 300 base pairs in length. And you just collect a ton of them, and you sort of bootstrap them together, and you build assemblies. And it's a really robust approach, and it's a really inexpensive approach at this point. But the sort of fatal flaw you can come up with in an environment like Antarctica is that you're getting life at its limit, but you're also capturing life optimally preserved. And so you can get these really short fragments of ancient DNA in the environment. And so ancient DNA is DNA that has been released from a lysed cell that has died, and it is now just sort of free-floating in the environment. And whereas in kind of a normal environment up where we live, those little bits of DNA will probably be gone, here they could still be sticking around. And one of the things that I was really interested in was testing these theories of long-term cell survival. So the Antarctica dry valleys are perfect because there used to be these big glacial lakes in these. So you see here are all the dry valleys. They sort of run across, and there used to be these enormous, much larger at least, much larger lakes. There's debate about how big and how long they were there. But the water stands were much higher, and they've retreated now down to these small lenses of ice-covered lakes. So they're perennially frozen at the top, but you've got liquid water underneath. And it's wild, these microbial mats that you get growing at the bottom of these lakes. And they have these beautiful conical structures, and they're just really amazing. But it's the bulk of the biomass in this environment. There's no, plant life. There used to be, in Antarctica, there used to be ferns and all kinds of things, but since, microbial biomass dominates. And so what you can see is in these lakes, the current lakes, you get these microbial mats, and then you get very, very similar looking features that are buried in perch deltas or just in other places up along the hillsides. So as you go up in elevation. And some of these are beneath bedded ash, which is terrific. You can also radiocarbon date these. And one of the things that we were interested in doing was testing these theories of long-term cell survival. Because one of the main ideas is that if you're going to stick around for a long time, if your climate conditions are going to change, you're going to hunker down, you're going to form a spore, you're going to shut down all your metabolic activity, and you're just going to wait. But one of the problems with that is that you're going to accumulate spontaneous damage to your genome over time. So you're going to get these oxidative damage, hydrolytic damage, you're going to get these nicks and these breaks, and eventually you may not be able to recover from that if your genome gets damaged enough. Because again, no metabolic activity means you have no DNA repair. So your genome is just your genome, and it could be really shoddy by the time the conditions can turn. And so it could potentially affect how we think about life detection strategies, in these places like Mars, are we going to find life that's just hunkered down, or are we going to find life that's actually doing some sort of low-level metabolic activity? But one of the things that we wanted to do for this study is really focus on these third-generation sequencing approaches because they do two things for us. Here are some examples. Well, let me tell you first the two things that they do. One is they're sequencing native DNA, which is great. So they're doing these single molecule sequencing approaches. So you're not amplifying amplicons, you're actually looking at your exact DNA strand. So you don't have any sort of PCR bias issues that you have to worry about. And second is you can get these incredibly long reads. So you could get reads that are 70,000 base pairs in length, one continuous strand, which is definitely not from an ancient DNA sort of situation. I mean, the longest piece of ancient DNA we've ever found, and it was in Antarctica, is up to 1,000 base pairs in length. Most DNA is really highly fragmented, kind of in that 300 to 500 base pair in length range. But if you get something that's like 10,000 base pairs long, that has definitely come out of an intact cell. So this is a cool way to help interrogate this question. And so I'm going to tell you about these two new third-generation sequencing approaches. One is the PacBio. So this is a new platform from Pacific Biosciences. There are a few dozen of these around. You use these tiny little zero-mode waveguides, so you get your one single strand of DNA, your single molecule coming in. And these are smaller than the wavelength of light. And you come and you've got your single polymerase, and you have these fluorescently tagged bases, and you can get, again, these really long reads. And one of the things I love about this is that you can see chemical modifications to the bases. So instead of saying, here's your A, T, C, G, you can actually say, oh, this is a cytosine, but it's been deaminated, or this is a methylated. You can see these little changes in the functional groups, which is great because one of the sort of most common forms of DNA damage is a deamination event, that 3 of the bases have amine groups, and you can knock those amine groups off. And so we can now use this to look at actual DNA damage along those strands. Again, it's native DNA, so that's neat. But actually, the thing that you guys will think is cooler is the Oxford Nanopore. So this is a different approach. So basically, you've got this tiny little protein nanopore. This is a couple nanometers across. And you put it in a membrane with high electronic resistance, and you get a potential applied across this membrane. And then you get changes in currents that you read. You do base calling four at a time. But you see these characteristic changes in current. As this gets ratcheted through, you get your strand of DNA, and it gets ratcheted through this tiny little nanopore. You put a bunch of these on a membrane. Oh, okay, another thing that we have to do is we have to make sure we get these very long reads. Standard methods, if you're going to try to use a vortexer with bead beating to extract your DNA from your cells, you could really shear up a lot of that DNA, and you're not going to get a 60,000 base pair read if all of your DNA has been sheared up in some sort of process as part of the extraction. So we've been developing these new methods to make sure we're being gentle enough, but at the same time using that microbial standard to make sure we're lysing everything so we can get a good look. But there's this massive miniaturization that's associated with this Oxford nanopore. So this little thing is a couple of nanometers across, and that's your protein nanopore there. And you array a bunch of them on this tiny little membrane here, and you put in some buffer, and you do your sequencing on this tiny little thing, okay? So this thing is 85 grams. It's 10 by 3 by 2 centimeters, and it draws less than a watt of power. And it's this big. And I should have brought mine. If my postdoc was not using it today, I would have brought it along and passed it around. It's half the size of a deck of cards. And so you could actually imagine sending this thing out as a on a mission. And so one of the things that we did while we were out in Antarctica is we were actually doing sequencing in the field with this tiny little thing. We had to use some hand warmers, essential lab gear, to make sure that it did not get too cold. But amazingly, even in temperature conditions of -5°C, we were able to do sequencing right there in the field, with just our little Mac laptop and our Oxford nanopore. So this, I think, is opening up all kinds of possibilities for doing remotely telemetered biological data. I mean, there are so many parts of our own biosphere that we are only barely beginning to understand. The deep oceans, underneath glaciers, these deserts. And if we could put an instrument like this and develop the software to actually have that biological data being remotely telemetered back to us in our laboratories, I feel like it could just be this incredible revolution, kind of like image data did for us at the beginning of the space age. All of a sudden we could see these places in our biosphere that were completely inaccessible. We could watch them change. We could potentially do this at the sort of next level for biology. But of course, one of the really exciting possibilities is the idea of sending one of these things up into space. Has been done. This is Kate Rubins on the ISS back in September. We took it up and just did a quick experiment looking at mouse DNA, E. coli DNA, and then the lambda bacteriophage, and just looking at making sure it works. It works even in microgravity, no deterioration in performance at all, which was great. But so what about the idea of sending a sequencer to space? It's been thought about. So there are two sort of main ways this has been pushed forward in the last couple of years. One is the idea to look, of course, for nucleic acid-based life. And, this is a really tenable sort of idea for Mars. We had, a billion tons of rocks exchanged between the planets during the late heavy bombardment. We know that some of those rocks were not superheated. Ben Weiss's work from MIT looking at, Curie temperatures in these meteorites. And so there is this possibility that you could potentially have a shared ancestry. Of course, this is again going after extant life as opposed to signs of ancient biomarkers. This is life that may be in one of these RSLs on Mars, maybe in a lava tube someplace protected from the surface environment. But we've got, physical similarities. We've got lots of rocks back and forth, probably more of those coming from Mars, of course, to the Earth than going the other direction because of the gravitational well of the sun. But we do know that so maybe we're Martians, but we've got, of course, a lot of microbial resistance that, we've got these bugs that can survive in the cores of nuclear reactors. Maybe you could survive that journey and just kind of catch life from the next planet over instead of independently evolving it. But maybe we should just look. I mean, wouldn't we feel silly if we didn't just look and there's life like our life that's just right there? One big advantage is that you would get something that's probably as close as you can to an unambiguous signal from this. So as we think about other types of biosignatures, the more broad ones can be a little bit more difficult to interpret because sometimes there are abiotic processes that can lead to these same sorts of, especially things like morphology. But if you find a sequence, you can immediately pin that onto a phylogenetic tree. You can say this is a deeply branching organism. This is similar to life, but it evolved. It took a different path billions of years ago, and this is unlike anything we've seen before. And there's also the possibility of using, of course, nanopore technology. It doesn't necessarily have to be DNA or RNA. You could have TNA, XNA, and you could look at non-standard bases with this same technology. The other big thing that's been pushed forward is the idea of using this as a contamination detection. Because if you're going to be sending, say, a suite of 6 or 7 instruments on a mission, you're looking for life, it's a big problem if you bring life with you. And of course, we can't get these spacecraft perfectly clean. Curiosity had 30,000 spores that it took up there. That was the planetary protection limit. And then we started seeing these RSLs near Curiosity. And it was like, oh, maybe it's not high enough. But one of the main things is Bacillus homeless. This is a big thing that when they wiped down the spacecraft and they did, extractions of the DNA on those washcloths, that's what they found. But you would know that immediately if you had a sequencer. You'd say, oh, it's Bacillus. There's this load of Bacillus, in this environment. And you know immediately that's a common contaminant, and it's kind of the sort of thing you would expect to see. And then these are other types of bacteria, and this is even a fungus that are common space room contaminants. But these two arguments have been advanced as why not take a sequencer to space, especially if they're so tiny, and there is this possibility of shared ancestry. But one of the things that I've been really excited about thinking on lately is the idea of harnessing the power of sequencing to look for life that's not DNA or RNA based. So the idea is that, and this has been the big kicker for why would you, there's a lot of resistance to taking a sequencer to space because again, it's like, well, are you thinking outside the box? Like our life is not going to be like Earth life, and especially once we start thinking about ocean worlds, we're further away. But we could also potentially use sequencing and these oligonucleotides to detect patterns of binding chemistry. And this is without even presupposing any kind of molecular framework. So here's the idea, is that you could take these single-stranded pieces of RNA or DNA, say 35 or 40 base pairs in length. And these tiny little oligos, they'll curl up and they'll have this secondary and tertiary structure. And what will happen is if you take a library of millions of randomly generated pieces of DNA or RNA, some very small subset, less than 1%, but some small subset of those are going to bind with incredible specificity and affinity to any ligands, any kind of analytes. And the idea is that you could have this kind of functional oligo once it sort of curls up, and you could have target binding. And the original idea from this came from the world of cancer research. There's this high, lots of fidelity for the celic stuff. I know Steely's worked on this before too. There's this idea that you can take your random pool and you can have your target cell. Say this is like a breast cancer tumor cell, and you incubate, and then a bunch of the stuff doesn't bind. Some of it does, and then you can get rid of the stuff that binds to negative cells. And you can amplify it up, and you can go through cycles because you'll have a little bit of non-specific binding. And you can find a couple of these short strands of DNA that are going to bind to where your cancer cells are. And that's a nice way of figuring out where your cancer cells are in the body. But this sort of idea that I've been working on with some colleagues here, this is Heather Graham at Goddard, Andy Ellington, and Eric Anslin at UT Austin, and then Paul Mahaffey has also been helping us from Goddard. This would be the idea of kind of flipping that. And so instead of trying to do any sort of selection process and say, oh, well, these sorts of things seem to be associated with, these would be good biomarkers, these particular strands, it would be to take the entire library. So you have millions of these randomly generated things. And you could do you could have a module where you could do some, say, size selection. You'd have to do some things that you'd have to get your sample in order. But say you've got something like a compartment, and you can flood these millions of randomly generated things over your compartment. and some very, very small percentage of them would bind. And like even if you just have a mineral surface, you're going to have these things binding, but the sort of vast amount of information that you would have on the surface of even the most primitive cell that we have here on Earth would be very, very interesting. And then you could do this ligation and amplification process, and then we could look at detection. So going into those, so these are kind of like antibodies, you know, and they function in a similar way, but because they're made of DNA instead of protein, you can amplify the signal. So you can go from, you know, just say very, very small levels of, say, compartments in your sample. And that may be the case for ocean worlds, you know, if we're not looking at photosynthetic life, which of course dominates our biomass here. If we instead have chemosynthetic life and very much lower, you know, using chemical sources of energy, you may only have, say, a few cells per, you know, gram of water, cc of water you collect from a plume on Enceladus. But if you could take that signal and you could amplify it up using something like PCR, you could get this really robust signal so that you could read it out. And then you could potentially have sort of a trifecta, right? So you could go after shared ancestry if you send a little sequencer. You could go after contamination, but you could potentially get these patterns of binding chemistry that would be very interesting to look at. So they may not be a definitive biosignature, but they could be a biohint. For example, a sample that had like much, much, much more complexity than another sample might be a suggestion that this would be a sample that could have something more interesting going on. And one of the things is that you will be able to sequence those particular oligos that do bind. And so you could go back and you could say, oh, there are these really weird minerals that we found, or there are these really weird simple organics that we found in this sample, you know, using our mass specs or using some of these other instruments. But you could go try to replicate that in the lab, and you could, you know, do binding with the same set of, that same random library, and you could say, oh, okay, definitely this is what it is. They're binding to these organics, or they're binding to these weird minerals. Or you could say, no, it's not, doesn't seem to be that. So anyway, this is like very, very, very early sort of discussion stage we put in for our Picasso grant. We'll see what sort of happens with that. But so you could potentially, again, like have another reason for sending a sequencer. You could kind of have this chemometric approach that would give you these patterns. And of course, if that gets too complicated, you could always do something like a microarray. Because maybe it's a little tricky to send a sequencer all the way out to Enceladus, particularly if you're doing it within the context of a low-cost mission. But these little chips, you know, about the size of a microscope slide, these microarrays have been used in genomics for a long time. You put lots of identical copies onto a spot, and so you could have the reverse complement strand for, you know, your different oligos that you're using. And you could have all these spots, and then you could just have a fluorescent probe released upon binding, so you could just kind of flow out after you do the amplification process onto one of these microarrays, and you could just optically image it. And, you know, send back this pattern, you know what's in every spot, and you could say, oh, well, this is the pattern for this sample, and this is the pattern for that sample, and, you know, you could do it that way. Or you could use an electrochemical microarray, which, like kerosene blue, it's another idea of using a, you have these redox reporters that help. And anyway, so there could be different mission opportunities for this kind of thing. You know, Europa, Enceladus, Mars. I'll always love Mars, closest to my heart. But I think, yeah, so I will stop there. I think I might be a little over time. But just to say, you know, here's my little lab at Georgetown, right down the road. And it's just so great to be in Washington with folks like you guys and these wonderful scientific institutions. And thanks so much for having me, and I'd love to take some questions if there are any. Yeah, sure.
The extremophiles, have you categorized them into archaea, bacteria, and eukarya?
Yeah.
What makes it good?
Oh, well, so you can have extremophiles of all three. In these particular environments that I've been looking at, the hydrothermal sites seem to have a little bit more on the archaea. The more lacustrine sites seem to be more heavily dominated by bacteria. But you can have extremophiles, like even eukaryotes, like in the Rio Tinto, which is this really acidic river, crazy place out in Spain. There are tons of like super extremophile eukaryotes there, which is sort of strange because you usually think, you know, kind of more simple things tend to do better in these super harsh places, and the eukaryotes tend to be more mainstream. But that was a cool result that was reported in Nature a couple of years back. That's a good question.
So your acid lakes, are they acidified actually by the metabolic processes in the microorganisms?
Well, so I mean, that's the real question, you know? Like, I mean, there's got to be pyrolysis going on too, and so not all of this, I think, is microbially generated. But I do think you could get these local pockets where you get more acidity from these microbial communities. And that's one of the things that we're trying to do now, is to do kind of like a more in-depth kind of, you know, survey where it's not just these bulk samples, but looking at the specific layering and comparing that with the mineralogy and try to do that at a higher resolution. Yeah, sure.
So, this analogy to the Australian soils is very interesting. But one of the real distinct characteristics of the Gale crater finds are this very high percentage of amorphous material, particularly amorphous material that's rich in sulfate itself. So, do you know of any terrestrial environments in which there's amorphous sulfate? Do you have any sense of how that might be forming? Some of us are speculating that the amorphous sulfate itself might be an abiosignature in as much as there's a lot of energy or crystallization associated with forming jarosite or copiapite or any of the other sulfates that occur, and it seems very odd. Or is it a sign of low water activity? Is it just a temperature thing? What's going on with 30 to 40% amorphous material?
That's crazy.
That's really different from Earth.
Yeah. I think that that's fascinating, and I wish I had an answer. I mean, it's just, I think that's one of the big mysteries, is trying to figure out these phases. And it may be water activity. I mean, of course, these environments on Earth are super saturated with water, and it'd be interesting to, you know, sort of see if we could run the experiment in a different kind of place. But I don't know of any places that have anywhere near those levels of amorphous sulfur and sulfates. So, I don't know. You should keep working on it.
I have a quick
Yeah,
question.
Stanley, sure.
Part of your using the array-based technology is your extraction protocol. Do you have any thoughts on that?
For using, for the nanopore for shared ancestry, or for the...
Or
Yeah.
using it as a
Well-
biosignature detection. Your extraction and then reading is going to be an issue.
Yeah, well, especially for the last one, you know, the sort of instrument concept. And the way we got around that with our little Picasso proposal is we said, oh, you don't need all the components. We're going to skip that one. We're just going to, you know, focus on the ligation and amplification and then the detection. But it is tricky. And I know that there are, even just for things that are, you know, Earth-based, there, like, Oxford Nanopore has a new little thing called Voltrax, which does some really nice, I wish I had a video. It has these little bubbles of fluid, and it moves them around, and you can even like spell out things with it, and it has, you know, heating that comes up and down. So there are these companies that are trying to come up with these kind of more commercial, off-the-shelf types of things for DNA extractions. And there's some progress in microfluidics in this front, but I think it is really tricky, and especially what we were talking about earlier with like super salty samples, or if you've got humics or you've got iron. I mean, you could have a lot of inhibition that comes out later. And, you know, we typically, if we've got super salty samples, we sometimes, you know, spin them down and do a wash process to try to get them into like a better range.
I think with the icy world, you have the opposite problem. You have to concentrate up what's there.
Yeah,
and
you may
it's
have
already
to.
in solution. You
Yeah.
can't concentrate it.
Well, or you could take enough of it. Maybe if you had something like this, you could still just do it. One of the things that we've been thinking about kind of in this very, very early thinking development stage of that instrument is to do this thing called a proximity ligation assay, where you're actually, instead of trying to bind cells or compartments by size sorting, you would actually do it all in solution, where the way it would work is you'd have your two oligos, but you put a 3' and a 5', like a little primer on each one, and then you'd have another one. And then the way it would work is some of those would bind to the same analyte, and if you had them in proximity, you could use a little splint, and then you could only amplify when you had the 3' and the 5'. So you'd have to go across, and you would only amplify the things that are there together. And then you could look at those. So that would allow you to do it in solution as opposed to, and then still not have to do like a wash process. But it's tricky, and I'm sure you probably know this more than anyone having thought about it. But it's, I don't know, that's like a lot of... Yeah.
Any other questions? So let's thank our speaker.
Thank you, everyone. This was great.
Thank you.